In this section, we're going to define mutations, discuss the types of mutations, and how we can differentiate the types of mutations and their different locations on the genome. There are different forms of genes that are called alleles. So if a gene codes for a certain thing, let's say it codes for your hair color, you know that there are different hair colors. So the gene itself is going to code to one protein. This one protein has more than one possible form. These different forms are going to cause the phenotype, so the way that the protein represents itself or its function. And these different types of the same genes are called alleles. Mutations are usually reserved for instances in which an allele is known to have been newly formed. So mutations are usually present when cells divide as a result of the replication process that we discussed earlier, or as a result of chemical disruption, which we also described earlier. However, most mutations take place when alleles are newly formed. A mutagen is an agent that causes a heritable change in the DNA sequence. So the key here is that the mutation should be heritable. So if a mutation happens, let's say, that causes a discoloration in the skin, in your skin cells, these are not part of the cells that your future children are going to have, because these are going to be reserved in gametes. So that this is not a mutagen that has caused a mutation in your skin. A mutagen has to cause a heritable change in the DNA sequence. The particular set of alleles for all the genes carried by an individual is called its genotype. So if you collect all the allele sets, and we know for each gene we have two alleles, one from your parental cells and one from your maternal cells. So you have the genotype is the collection of all the alleles in all the genes that are carried by yourself. Another definition that we need to go through is what a wild type is. A wild type is a phenotype. It's used to designate a standard genotype for the use as a reference in breeding experiments. So let's say that the quote-unquote regular gene is the one that always shows up. So let's say all bears are brown. So the wild type is brown. If there is a mutation, you could have, for example, a white bear. However, the wild type is the brown, and that's the dominant, and it's the more standard genotype that represents itself in the population that you have. A phenotype are the physical attributes or traits of an individual that are the consequence of a given genotype. So we know that we're carrying all these genes, and these genes manifest themselves into different traits in our behavior, in our bodies, in the way that we look. So the phenotype is how these genes represent themselves in actuality. So you have, for example, a phenotype of somebody's height, or a phenotype of somebody, the way they look, or the shape of their eyes. So all of these are phenotypes. They are not genotypes. The gene, we have to say a gene number that codes for a certain protein. However, the phenotype is how this gene manifests in order to show us a certain trait in a person. Each organism is either a haploid or a diploid organism. Haploid cells have half the amount that diploid cells have. So let's take humans as an example. Creatures with diploid cells have two sets of chromosomes. Each set comes from each parent. So humans have 46 chromosomes. 23 chromosomes come from each parent, and each parent has a chromosome that's homologous to another chromosome that's coming from the other parent. So for example, let's say we have the gene for hair color, and the dominant is black, while the recessive is blonde. So if we have black, which is the capital B representation here, is dominant gene, it will be in one of the two homologous chromosomes coming from one of the parents, while the other gene that is homologous to it comes from the other parent. So let's say it's the lowercase b, which is the blonde. So you have one gene coming from one parent, it's homologous chromosome coming from the other parents. They both code for the same gene, so they both code for hair color in this instance, but they don't have the same code, because this one codes for black hair while this one codes for blonde hair. Again, they code for hair color, but they are different sequences, and each comes from each parents. So each diploid organism, including humans, has two alleles, which are the homologous chromosomes. Each allele comes from each parent, and so you have two copies of the same gene in your cell. If the two alleles have the same gene exactly, so they have exactly the same code, so we have two black haired genes, that is a homozygote cell, or a homozygote gene. So if we have two blacks or two blondes, this is an example of a homozygote cell for a certain gene, in this case hair color. Heterozygote cells have two different genes or two different alleles for the same gene. This example is heterozygote. So a recessive mutant allele is one where both alleles must be mutant in order for the mutant phenotype to be observed. As you remember, each gene has a phenotype and a genotype. So some genes that you get, the genotype is the sequence of the gene that is encoded in your genome, while the phenotype is the expression of that gene or the characteristic that you get from that gene. So the black hair color in this example is the phenotype. The blonde hair color in this example is the phenotype. However, the two recessive genes, the lowercase b and the lowercase b, are the genotype. The large case b and the uppercase b are the genotype. So in order for the mutant allele to be observed phenotypically, so for the blonde hair to show up, you have to have two lowercase b's. You have to have a homozygote cell with both blonde genes. If you have black hair, it could be either that you have a homozygote with two uppercase b's, or you could have a heterozygote with an uppercase b and a lowercase b. So phenotypically, these two are the same. However, genotypically, they are not the same. A good example are silent mutations. So we could have a silent mutations, which is a mutation that does not show up phenotypically. So we can carry a certain mutation. For example, thalassemia is a very common mutation that is carried in the Mediterranean region, including Jordan. A lot of people carry the gene for thalassemia. However, it's not expressed in their blood. They are not sick. They don't have thalassemia. However, if you have a homozygote with the two thalassemia mutations, the phenotype is going to be observed, so the person is going to be sick with thalassemia. A good example also of genetic mutations for recessive genes are sickle cell anemia. As you know, red blood cells have a double concave shape. Once there is a mutation in the hemoglobin protein, they get a sickle cell shape, so they look like a sickle. That's the device used for cutting wheat. So if this mutation shows up phenotypically, if both the alleles that we have the genetic mutation, so it's a recessive mutation. However, there's an outside selection mechanism for this mutation, because it turns out that people who have sickle cell anemia do not get malaria, because malaria only infects people with healthy red blood cells. So what happened is that people who had healthy red blood cells got infected with malaria in Africa, and they died as a result of getting malaria. So people who had sickle cell anemia did not get malaria, so the number of people who have the mutation has increased by using an external selection factor like malaria. This is a picture representation of the recessive and dominant mutations, as we just mentioned. So we have the genotype on top and the phenotype on top, on the bottom. And notice that we are talking about diploid cells. So we have a copy, two copies from each gene, a copy from the father and a copy from the mother. So genetically, if we have two wild types, we're going to end up with a wild type phenotype. If you remember, we said wild type is the prevalent standard phenotype or genetic structure that represents a certain gene. So if our mutant is dominant, the mutation is dominant, and the wild type is recessive, we have a heterozygote diploid genotype that would result in the representation of the mutant gene in the phenotype, because it is the dominant gene. If we have two dominant mutations, that is going to represent itself phenotypically with a mutant phenotype. If we have a mutation that is recessive, then the wild type is going to be dominant, causing the wild type to show up as the phenotype. So you would need two recessive mutation for the mutation to manifest itself in the phenotype. There are three different types of mutations. There's the missense mutation. In this missense mutation, the sequence of amino acid is different because some of the amino acids are substituted. So let's say you have a GCGCA, for example. This GCGCA is then replicated into a GGGA. So that is a missense mutation. The second type of mutation is the nonsense mutation. Here in this type of mutation, an early stop codon is reached. So as the mutation is being translated into a protein, a stop codon is found in the middle of the gene, so the protein is translation is terminated and the whole protein does not get translated. There's also the frame shift mutation. This mutation is the result in the shift of the reading frame. If you remember, each codon has each amino acid has three bases coding for it. So if we have a shift in the framework of translation, the three bases are going to become different. This is going to cause a result of having a continuous missense mutation as a result of this frame shift. The most minor effect from a frame shift mutation takes place when 3 or 6 or 9, so multiples of 3, because for each amino acid we need 3 bases. If the frame shift takes place by 3 bases or 6 bases or a multiple of 3, then we only use 1 or 2 amino acids. However, if the frame shift is 1 or 2 or additions of 3 to these two numbers, then we are going to get a complete frame shift, which could result in translating a completely different protein. As we mentioned a couple of slides ago, cells can result from division. That division can either be mitosis, where the number of chromosomes in the paternal cells is preserved, and we gave an example of humans who have 46 chromosomes, so they have 23 chromosomes from each person. That is double the number of chromosomes that we get if we have the cell division of meiosis, which results in half the number of chromosomes in the cell. If we have meiosis and we have 23 chromosomes for humans, that means that we have created cells that our bodies are going to use for procreation. So this 23 chromosome cell needs to fuse with another 23 chromosome cell in order to make a complete cell that can further divide and make a fetus. So in meiosis, we are usually creating gametes. Gametes could either be ovum in the females, or they could be in sperm in males. However, if we are talking about mitosis and complete cells that can deliver different functions, we are talking about the creation of somatic cells. Most cell divisions that takes place in the body are mitotic, and most cells by far in our bodies are somatic cells and not gametes. The danger in creating gametes in meiosis is that this is where mutagens are effective, as I mentioned at the beginning of the slide set, because any mutation that takes place in the gametes can be passed down to future generations, and this is where the danger of having a mutation in gamete formation is dangerous. Usually, if we want to understand in experimentation the wild type and dominant and recessive genes, we need to have a homozygous wild type so that the gametes that result are both the same, which would be easier for future genetic studies. As we mentioned, we have mitotic cell division and meiotic cell division. In mitotic cell division, we have two collections of chromosomes resulting in 2n number of chromosomes. N represents the haploid number of chromosomes. In the case of humans, it's 23 chromosomes. And before the cell divides in mitosis, we have DNA replication to double the amount of DNA material within the cell. So we'll have double the 46 chromosomes. That is 4n, so it's 4 times the 23 chromosomes. Afterwards, we get the mitotic apparatus that where the chromosomes align at the middle of the cell. This is where recombination and crossover takes place, by the way, so that genetic information is exchanged between two homologous chromosomes. After recombination takes place, the cell divides into two cells. Each of these cells has 2n chromosomes, so in the case of humans, it has 46 chromosomes. In the case of mitotic cell division, we have paternal strands and maternal strands. Then we get DNA replication. Here also we get a 4n chromosomal cell. We also then get the mitotic apparatus where homologous chromosomes exchange genetic material in recombination. This is what we discussed in the previous slide set, recombination. If you notice here, you get two complete chromosomes, one paternal and one maternal. However, you can see that there's a discrepancy in the color of the chromosome, so you get maternal coloring in the paternal chromosome and paternal coloring in the maternal chromosome, and that is the result of recombination. Here the cell divides into two daughter cells, each having 46 chromosomes. So up to here it's exactly the same as mitosis. However, afterwards, because we have reached the second phase of meiosis, each of these two cells is going to divide further into two other cells. However, in this case, there's no replication before cell division. So each of the daughter cell here will have half the number of cells in the paternal, have the number of chromosomes, in the paternal cell, getting 23 chromosomes in each cell and creating gametes. Also notice here that we have different color chromosome combination, meaning that the ones that only have one color come from either the father or the mother. However, these that have two colors in them have a more genetic variation due to the process of recombination. So if we wanted to study whether a certain trait is a mutant or a wild type, it's better to mate homozygote whatever species we're using, whether it's flies or frogs or whatever. It's easier to mate mutants that are homozygous with wild types that are homozygous, because in this case we know that all gametes are going to have the same genotype in each case. So if you combine the dominant gamete with what you know for sure is with another homozygote gamete, you're going to get a dominant phenotype with a combinatory genotype. It's yellow because the dominant is yellow. However, genetically it's different from this mutant, and it's different from the wild type because you have one dominant mutant allele and one recessive wild type allele. If we further divide this cell into gametes, you have them mate with one another, you're going to get a dominant and a recessive in one case, and a dominant and recessive with the other. If you see the different possible combination, you're going to get a dominant and a dominant, giving you a mutant phenotype, a dominant and a recessive, giving you again a mutant phenotype, a dominant and a recessive, again resulting in a mutant phenotype, and finally a recessive and a recessive resulting in a normal wild type. In this example, the mutant is recessive, so we need two alleles of the mutant to get the phenotype, and the wild type is dominant, so we only need one of the two alleles to have the wild type in order for the wild type phenotype to be expressed. So if we divide these cells, all the gametes from each of these is going to be pure. You're going to get all mutant from the mutant example and all dominant from the wild type. If these are mated with one another, all of their progeny is going to be a dominant wild type with a recessive mutant leading to a wild type phenotype. If this is further divided, you're going to either get a recessive mutant gamete or a dominant wild type gamete. In this case, either you're going to get a recessive mutant with a recessive mutant, which is going to lead to a mutant phenotype, or you're going to get a recessive mutant with a dominant wild type, a recessive mutant with a dominant wild type, or a dominant wild type with a dominant wild type, and the last three cases are going to result in a phenotype that is normal, or what we call the wild type phenotype. The best model to study eukaryotic cells and the genetic structure of these eukaryotic cells are yeast cells, and that's because they are not prokaryotes, they're eukaryotes, but they're the simplest type of eukaryotes. Cell division takes place quickly, which is a must. You don't want to wait for years for a species to replicate. And the cells are not very complicated themselves in terms of the amount of genetic material, etc. So if we get a recessive mutant, and note here that recessive genes are represented with a lowercase letter, while dominant genes are represented with uppercase letter. So here the mutant is still yellow, however in this case it's recessive. So all the resulting gametes are going to be a recessive mutant gametes. The wild type is dominant, however it's homozygote dominant, so you get all the gametes are going to be the same. If these two mate, we're going to get all their progeny is going to have a combination of a recessive dominant mutant with a dominant wild type, resulting in a dominant wild type genotype. If this generation mates with itself, that means in each case we're either going to get a recessive mutant or a wild type that is dominant. Again, you're going to either get a recessive mutant with a recessive mutant resulting in a mutant phenotype, a recessive mutant with a wild type that is dominant, getting a normal wild type phenotype. Again, you can either get a dominant wild type with a recessive mutant or a dominant wild type with a dominant wild type. All of these are going to be a wild type normal phenotypes. A type of mutation that is heavily used in research is called conditional mutations. So these mutations show up phenotypically only when certain conditions are met. A good example of conditional mutations are temperature sensitive mutations, which depend on the stability of proteins at different temperature values. So for these conditional mutations, and more specifically for temperature sensitive mutation, you have two temperatures. The non-permissive temperature, here the mutant phenotype is observed. So if the mutation is present and we are at the non-permissive temperature, the mutant phenotype is observed. If we are at the permissive temperature, then the mutant phenotype is not observed. So if we reach above a certain temperature, the mutation is going to manifest itself phenotypically. However, if we remain in the permissive temperature, then the mutant phenotype is not going to show up phenotypically. So as an example of conditional mutation, we have if we have yeast in liquid cell culture, we add a mutagenic material that's going to cause mutations in the yeast cells, and then we divide these cells into smaller amounts called aliquots. Afterwards, we can plate these different aliquots onto different agar plates, and we keep this incubation at 23 degrees Celsius for a few hours. Then afterwards, these plate replicas are kept at different temperatures. So some of them are kept at 23 degrees centigrade, where the others are kept at 36 degrees centigrade. So if you notice at 23 degrees centigrade, all the colonies that result from bacterial growth are remaining in the plate, where at 36 degrees centigrade some of the colonies are missing. This is because we have a temperature sensitive mutation. In this mutation, what happens is that once we reach the permissive temperature, the mutation manifests itself and it leads to stopping the growth of the bacterial cells, which results in the disappearance of the colonies. However, at 23 degrees, we have the non-permissive temperature, so the mutation is not evident for us, and all the colonies numbers remain the same. Here we can see that if we mate wild type mutation with other yeast cells that have a type of mutation called CDC 28 and CDC 27, that you can observe the phenotypic differences either through the shape of the cells or the color of the cells from the standard wild type. You can see that the CDC7 mutants look more like the wild type than do the CDC 28 mutants. For studying whether certain mutations are on the same gene, the same location, or at different genes, there is a test called the complementation test. And it depends on restoring the wild type phenotype by mating two different mutants. So the major idea is that if we have two recessive mutations on the same gene, then the diploid organism that is heterozygous for both a mutation will exhibit the mutant, because neither allele is going to have a functional copy of the gene, because it's a recessive mutation on the same, two recessive mutations on the same gene in a diploid organism. That means we're going to get the recessive mutation. If the mutations are on separate genes, then the heterozygotes carrying a copy of each gene, since the gene is recessive, the mutation is recessive, then it will not show the mutation phenotypically, because the wild type allele is going to be present and it's dominant. It would be better explained later on in the figures. So here we have type A mutant and a type alpha mutant in two different plates. You mate the haploids of the opposite mating types. These carry different recessive temperature sensitive CDC mutation. So the question here that we're trying to answer with this test is whether the genes that are mutated are on the same chromosome or the same location, or if they are at different locations. We are not trying to see whether there's a mutation or not, because we know for a fact that there is one. The question that a complementation test tried to answer is whether these genes are lumped together, the mutations are in the same location or at two different locations. Also keep in mind that the mutations are recessive. So if we have all these are yellow, so all of these are mutated. This is the first thing we need to take to keep in mind. However, this is a CDCX with a CDCY, and this is a CDCX with a CDCZ, and we're trying to see if the location of these two are the same and the location of these two are the same. So if the location is different, what happens is that if we repeat the experiment that we just explained, we'd have diploids that have temperature sensitive CDC phenotype in both cells. However, if we go to the non-permissive temperature, what happens is that in the first plate, the phenotype of the mutation does not show up, but it does show up in the second plate. What this indicates is that even though we know there's a mutation, phenotypically it didn't show up. So these two are recessive and these two's location are different. However, since the phenotypically this showed up, this means that because these two are recessive, then they are present in both alleles, meaning that they are in the same location. So the CDCX and CDCY are on different genes, and because it's a recessive mutation, phenotypically the wild type shows up, and the cells grow normally at the non-permissive temperature. However, in this case, phenotypically the mutation shows up, even though it's a recessive mutation, meaning that the mutation is present on both allele, which indicates that since the mutation is present on both allele, that there is no case where there's a dominant gene in the paternal genes, meaning that both mutations are on the same location, because there is no wild type showing up in the plate. So these mutations that are more than one mutation that are present in the genome have a lot of effects depending on their location and the relation of the two mutations one to the other. So we have, for example, mutations that take place in a biosynthetic pathway, and we talked about some of these pathways when we talked about, for example, protein synthesis or DNA replication, or when we talked in your biochemistry class about the Krebs cycle or whatever. You notice that there is a series of proteins and enzymes that do a certain job. So if there's a mutation in let's say two enzymes that are part of the same pathway, what happens is that we need to tell if the mutation is in enzyme A or enzyme B. So if we don't get this final product that here is represented by the number 3, it doesn't mean that there's a mutation in this enzyme. The mutation could be in this enzyme, because if we do not obtain the intermediate, then the amount of the reactant here is 0, so we're not going to get anything here. So in order to know whether the mutation is in the first enzyme or in the second enzyme, because this is a reaction that is a pathway that's catalyzed by two enzymes, what we need to do is test the amount of the reactant, the initial reactant, the product of enzyme A, which is also the reactant of enzyme B, and the product of the whole reaction, which is the final product of enzyme B. If there is no change in the amount of a reactant here, we can tell that A is not catalyzing this reactant number 1, so we are not getting any product. We can also test the amount of this intermediary product here. If the amount here is great, meaning that 1 has been catalyzed into 2 by A and there is no product 3, that means that even though the reactant is present, we don't have a product, meaning that enzyme B is not doing its job. Also, when a double mutation takes place in a signaling pathway, there are more than one possible scenario. The first one is the one illustrated in front of you. So you have the enzyme A, it gives repressed reporter expression, while B gives the constitutive reporter expression if there is a mutation. So what A does is increase the reporter expression. If there's a mutation in it, that would repress it. B represses A, which would reduce the reporter expression. However, if there's a mutation in B, that would increase the level of A and hence increase the amount of the reporter. So what happens in the case of a double mutation, what happens is that if this is mutated and this is mutated, then the reporter is going to be less expressed and repressed because A positively regulates the reporter expression and is negatively regulated by B. So if there is a mutation in A, we're going to get a repressed reporter expression. If there's a mutation only in B, we're going to get an increase in reporter expression. If there's a mutation in A, we're going to get a decreased reporter expression. So these combinations can help us track whether the mutation is in both proteins or in one of them. Another example, similar to the one before, is that if a double mutation in A and B causes this reporter to be continuously expressed, because B is supposed to deregulate the expression of this reporter, so it's going to stop it, and A regulates the expression of B. So if we have a double mutation, this is going to cause this reporter to keep expressing itself. The same thing. If A is not present, then B is going to suppress the reporter. If A is present in very low amounts, then B is not going to, then if A does not suppress B, then B is going to suppress the reporter. If A suppresses B, then B is going to allow the reporter to be expressed. So we need to basically see the effect of each enzyme in the pathway separately, or each signaling molecule separately. Some mutations are called suppressor mutations, and these depend on whether the mutation is present in both alleles or not, and how it affects the genotype and the phenotype. So if the genotype for the dominant wild type is dominant, we have both proteins binding to one another, causing them to accomplish whatever function they have to do. If there's a mutation, a recessive mutation in one of the alleles and no mutation in the other, then they can bind, however loosely, and that is not going to allow them to accomplish their function. If the mutation is in the other allele, then they are also not going to bind well, which is going to cause a problem in the phenotype, because if a lot of binding events cause a lot of pathways to be triggered, as we talked about in this course and you've taken in other courses. So if this binding event doesn't take place, then that could stop important biochemical activities. The interesting part, and what we wanted to mention in this slide, is that a possible double mutation, a mutation in each of the two alleles, could lead to a mutation where the two can bind to one another, even though they do not have the same genotype as the wild type. And if these two bind to one another with the same binding affinity as this wild type genotype, then the proteins can accomplish their function, and that mutation is suppressed because the proteins still can accomplish their function. Synthetic lethal mutations have a very dangerous effect because having two mutations that are consecutive or on the same gene can result in detrimental effects. Synthetic mutations can also reveal if there are non-essential genes whose proteins are redundant, so you have more than one protein that can participate in a pathway to produce, for example, a certain cell component or as part of a signaling message. This can better explain what we're talking about in synthetic lethal mutations. So the first type is completely the opposite of the suppressor mutation. So if we have the genotype wild type, then you have both proteins binding together pretty well. If we have a defect or a mutation in one of the two alleles, this could lead to partial binding, so a partial defect. If we have a mutation in the other allele, that could lead to a different partial defect and a partial mutation. However, if both locations are mutated, that's going to completely prevent the two proteins from binding and doing their function, which would lead to a severe defect. The other type of synthetic lethal mutation is when the wild type changes a precursor in large amounts. So we have what we just called redundant proteins. So these two accomplish the same function. So if one of them has a mutation, the second one can still produce the same product. The same thing goes if the second one has a mutation, the first one can still produce the same product. And these are present in biochemical processes that result in high demand components and products that are needed in biochemical reactions. However, the synthetic lethal mutation takes place when both alleles have a mutation, which completely prevents the synthesis of the target product, which is going to stop one or more biochemical pathway.